So hello everyone and welcome to today's IQ webinar. My name is Hongbin Yu. I'm from Boehringer Ingelheim Pharmaceutics in Ridgefield, Connecticut. So I'm very happy to co-present with Prakash Baladula from EMD Sereno, Marcus Wallace from Novartis, and Catherine Ball from AstraZeneca. The title of our presentation, the perspective on ADME of Therapeutic Proteins. And I know that many of you have heard of IQ or even participated for IQ-sponsored activities. So IQ stands for the International Consortium for Innovation and Quality in Pharmaceutical Development. Its vision is to be a leading science-based organization, advance innovative solutions to biomedical problems, and enabling pharmaceutical companies to bring quality medicine to patients. The mission of IQ is to advance science and technology to augment the capability of member companies to bring transformational solutions that bring benefits to patients, regulators, and the broader R&D community. So let's dive right into our presentation. Here is the presentation outline. I will start with a very short introduction. Then Prakash will go on to present on the current trends and the future perspectives of ADME of therapeutic proteins. After that, Marcus will present on the biotransformation of therapeutic proteins. Catherine will talk about biodistribution and PK of therapeutic proteins after Marcus. I will come back at the end to summarize our talk, and then Brooke Rock of Amgen will moderate the panel discussion after the presentation. So now let me start with the introduction. As many of you know, in the last few decades, therapeutic protein have become a very important part of pharmaceutical development and approval. And as you see in this picture, therapeutic proteins has an array of modalities and very diverse structure. I mean, the most common one is the monoclonal antibody, as you see on the, let me just put on the pointer here on the left upper corner. But in between over here, you see many antibody-based constructs as well as proteins. So there's many different flavors of therapeutic proteins. It's because of this very diverse structure of therapeutic proteins that led to many complexities and the challenges in terms of characterizing their ADME properties. Comparing to small molecules, they have very different absorption, distribution, metabolism, and the excretion processes with the small molecules. What makes problems even a little more complex, currently there are very limited regulatory guidance in terms of how to study the ADME properties of therapeutic proteins. In this context, the IQ, Translational and ADME leadership group has decided to form a working group to tackle this issue in 2019. So since that time, a great working group called ADME of Therapeutic Proteins group was formed. In the last 3+ years, we have had 40 members participating in this group, representing 20 companies. And this is a very much collaborative and scientifically driven group, and had a lot of fun working with this group. And we also have been very, very productive. So I'm very proud of the work that this group has done. So just give you a little bit of history of where we came from and what we have accomplished. So as I mentioned earlier, in 2019 or mid of 2019, the TALG, the Translational and ADME Leadership Group, formed this discussion group on the ADME of therapeutic proteins. The purpose was just to have identify the initial scope and the focus areas. But then there's so much interest among the team members within this discussion group, we quickly proposed to form a formal working group with defined focus areas as well as deliverables for this working group. That is at the end of 2019. So in 2020, I think that is the year of the pandemic started, and this year we had a lot of groundwork done. So we divided our work group into 4 subgroups, and we also had an IQ cross-company survey try to understand the current status of ADME studies of therapeutic proteins. Based on the foundational work that we did in 2020, so we started to write 3 papers, and we had 3 sub-teams working on 3 white papers. In the mid of 2021, we also got an invitation from DMD to publish our papers in a special section in 2022. So that was our goal. So we submitted our paper in 4Q of 2021. Then at the beginning of this year, we were very happy that DMD accepted our like a manuscript. We did some revision, and finally the work was published in the June issue of DMD. So today we are very happy to, through this webinar, to share the survey results that we have obtained from cross-company perspective, and also the insights from our publications. So we also hope to seek your feedback at the end of this presentation. So that is my last slide over here. I just want to share with you that we're very happy that we not only published 3 papers in the June issue of DMD this year. I think our work also is featured on the cover page of this special section on pharmacokinetics and ADME of biological therapeutics. So we are very happy, and I'm very proud of the work that we have done. And in the next few presentations, Prakash, Marcus, and Catherine will talk about the highlights from each of the 3 papers that we have published. So at this time, I would pass the stage to Prakash. Thank you so much.
Very good. Thank you, Hongbin. My name is Prakash Boladala. I'm at EMD Sereno in Quantitative Pharmacology Department. So one of the sub-teams in the working group has looked at the current practices of ADME of therapeutic proteins. In next few slides, I'll walk you through the results. Before going into the details, so just to provide a brief background on therapeutic proteins. Therapeutic proteins are engineered proteins that are intended for replace or repair the abnormal or deficient proteins in the human body. So as you know, that therapeutic proteins exhibit their effect by augmenting or blocking an existing pathway. And also last decade or so, therapeutic proteins has been provided many options for disease areas such as oncology, hematology, all the way to musculoskeletal diseases. Therapeutic proteins most often is a specific and selective, and it elicits the very limited or no adverse events generally. And also the DMPK and clinical pharmacology development for therapeutic proteins are relatively less complex compared to the small molecule drugs. Coming to the ADME of therapeutic proteins exhibit distinct ADME processes, and is different from the small molecule drugs, mainly due to the large size and in generally lack of cellular permeability, and also the difference in metabolic fate. For example, if you look at the small molecule drugs, the small molecule drugs produces a diverse metabolite or metabolic pathways, whereas in the therapeutic proteins primarily undergo the proteolytic degradation. So coming to absorption is an important parameter for therapeutic proteins that particularly administered through a non-intravenous route. And obviously, the pharmacokinetics of therapeutic proteins are affected by multiple factors, such as the bioavailability, anti-drug antibody formation, and also target-mediated disposition. The distribution is also a critical component, particularly to understand the efficacy and safety, because whether the therapeutic proteins is distributed into your target tissue, or it is distributing to off-target tissues to produce any unwanted effect. The metabolism and excretion is, as I mentioned previously, this is primarily undergoes the proteolytic degradation and renal filtration. Therefore, the primary objectives of our working group are really to understand the current practices of ADME data generated in the therapeutic proteins across the IQ member companies, and also to learn about the strategies followed in the member companies and to provide a recommendations on a best practices for conducting the ADME studies. So what we did actually, we conducted an industry-wide survey between March and May of 2021, and with a survey questions focused around the routes of administration, distribution studies, and also the data utility of these distribution studies, the characterization of active catabolites, and also the biotransformation, and in addition to that, the regulatory strategies and regulatory interaction experience among the participating companies. So we requested one collated response from each representative company. The results are all blinded and processed by IQ Secretariat, and they provided to us for further analysis. Simultaneously, what we did is we also reviewed the BLA applications that have been submitted to FDA from 2011 to 2020 and looked for all ADME data, particularly around the preclinical ADME data that has been submitted in these BLA applications. We were able to gather approximately 90 BLA applications and then looked at possibly ADME data in that application. If you look at this table here, the absorption, what we did here is absorption that includes the pharmacokinetics and bioavailability, but pretty much like all the BLA's applications had a pharmacokinetics data in it. And if you come to the distribution, there are a limited number of distribution studies been submitted to the in BLAs. And then metabolism and excretion, almost none to very few, depending on the modality type. So these are the survey results. First of all, the first question is really focused: does your company conduct an ADME of therapeutic protein? What type of ADME studies you conduct for this therapeutic protein? If you look at this, green says yes, and then blue says no. Pretty much majority of the companies are conducting biodistribution studies, and also about 50% of the companies conducting the biotransformation or metabolism studies. Very limited number of companies conduct the elimination routes, particularly on this therapeutic protein. And the another question we asked is the mechanism of absorption, but very limited number of companies responded yes for this particular question. That primarily we meant the mechanism of absorption could be related to the bioavailability. When you're having intravenous, most of the intravenous administration of therapeutic proteins, the bioavailability may not be conducted. That could be a response from the company. So the next question, what we asked is administration routes and non-clinical species that is being used in the pharmacokinetic assessment. If you look at the left-hand side pie chart, the majority of the companies still develop a therapeutic proteins with an intravenous route of administration. More recently, there is subcutaneous route of administration is also being evolved. The right-hand side of figure, the in vivo models that are being used for the PK assessment of this therapeutic protein is primarily the wild-type mice, FcRn mice, and non-human primates is primary species being used for pharmacokinetic assessment. And the table here is consistent with what we have seen from the survey results. So I have provided here as a representative waiver of 2018. If you look at the dosing route, basically it's primarily an IV, but although there is a subcutaneous is being evolved more recently. In the PK species, if you look at this, is mouse and monkey is the predominant species that is being used for the pharmacokinetic assessment. So the next question, what we asked was, does your company perform a biodistribution studies? If so, would you conduct these studies in a preclinical species, or would you also conduct in humans as well? How this data is being utilized in your drug discovery and development program. So if you look at the top of the row here, pretty much like approximately 70 to 80% of the companies responded, they do conduct the biodistribution studies. But about 50% they responded, yes, they would conduct the biodistribution in human studies. So how this data being utilized, particularly around the preclinical data, they are being primarily utilized to build a translational PK/PD modeling for these therapeutic proteins. So the next, the follow-up speakers, but Marcus and as well as Catherine will provide more detail how really this biotransformation and distribution data that is being utilized are in the overall development, discovery and development process. So if you look at the right-hand side, so we have provided that the survey results in the, provided from the BLA application, where you are seeing the distribution studies are limited, but using the radio-labeled material, either iodine 125 or indium 111, and rarely you can also use the tritium label to study the distribution of the therapeutic protein. The main discrepancy from the survey versus the BLA applications, what we are seeing is primarily the distribution studies are being maybe conducted in the early discovery and prior to candidate selection stages, or prior to IND stages, so where the distribution studies are not really submitted for in the BLA application, rather than primarily used for the translational PK/PD modeling to project the efficacious doses or human or predict the human dose. The biotransformation data, like this next question is really focused on the biotransformation data. So how you utilize the biotransformation data? Do you conduct the biotransformation, and how do you utilize this biotransformation data? If you look at this figure here, as from previous response, there is about 50% they conduct the biotransformation data. Among the biotransformation data, when pretty much majority of the companies do use the biotransformation data, particularly to understand the clearance mechanism and to address if there is a PK/PD disconnect, and also in safety to address some of the safety information. So the last question is really asking the regulatory strategy within the company. So we have 3 questions in this the major question, main question. Does your company have a regulatory strategy for ADME of Therapeutic? Pretty much the majority of the companies said no. And then also the next question focused really whether you have any regulatory interaction experience, and also which led to a conduct of additional ADME studies. Again, the majority of the first respondents said, like 73% said there is either no experience, or they have not conducted any ADME studies upon the request of regulatory agencies. So we also asked, what is your expectation on the need for further regulatory guidance on the ADME of therapeutic protein? If you look at this here, like majority of the companies stayed more, neutral. So they didn't say whether they needed any further guidance. But however, if you look at that, like the information or whatever the literature available at this time looks like reasonable from the survey point of view. So with that, I would like to the conclusion, and also we proposed future perspective. For therapeutic proteins that are not administered intravenously, absorption and bioavailability should be continued to be assessed in a non-clinical species along with the pharmacokinetics. It is also, as from a working group perspective, we see the value of conducting the biodistribution studies, and it should be a fit for purpose biodistribution studies, and the emphasis will be certainly incorporating the data into a translational PK/PD modeling for better understand the systemic and target exposures. So the need for additional biotransformation studies of therapeutic proteins mainly lies in the preclinical stages for choosing likely developmental candidate, and also addressing any disconnects, particularly on the clearance mechanisms and or PK/PD disconnects, and also explain the safety concerns and so on, but not necessarily for the regulatory submission package. So for conducting the elimination or exclusion studies, really you need to think at the size of the therapeutic protein, and also is there any specific population such as the renal or hepatic impairment population? How is that going to be affect the pharmacokinetics of the drug? It needs to be considered. So although based on the survey, the desire for additional regulatory guidance is very moderate, However, working group believes there is some further clarification in the guidance is required, while recognizing there is a significant diversity in the therapeutic proteins, and also addressing some of the low risk for this off-target effects of therapeutic proteins. So with that, I would pass on to my colleague, Marcus Wallace, for next topic presentation.
Thank you, Prakash. Thank you very much. Thank you. I'm very glad to present on behalf of the team our perspective on therapeutic protein biotransformation. The team members of my team are listed below, and they're really experts in that field. A lot of them are online today, and they will be available to answer your questions later if you have some. So let me start, give you an introduction. I think the biotransformation of small molecules is well understood, and also the impact. So we have a feeling for what drug modifications on small molecules, either enzymatically or chemically, can do, and what the impact on pharmacodynamics, safety, and efficacy is. For large molecules, it's a little different. They are perceived to just get broken down to peptides and amino acids, which are not perceived to be a safety liability. And I think some regulatory guidance even state that, as you see on the slide, that therefore further biotransformation investigations of these kind of moieties is not required. However, you have seen in the survey Prakash has shown before that about half of the companies conduct biotransformation studies for these proteins. And this is partly also because the profile has changed. I think at the time of the guidance, the prevalent registered proteins were monoclonal antibodies. As Hongbin has shown, I think the field has changed. We have now much more diverse constructs. We have bispecific fusion proteins, pegylated proteins, ADCs, and a lot of these new constructs, they actually undergo biotransformation in circulation, and therefore there's a clear need to do an assessment. And we will focus in the next couple of minutes to elucidate some of the most prevalent biotransformation reactions in proteins, meaning proteolysis, amino acid modification, and also biotransformation of tethered biotherapeutics like ADs and pegylated proteins. We'll talk a little bit about analytical methods and the impact of these findings on PK/PD and immunogenicity and safety. And last but not least, we will propose a decision tree, how we see decision tree which can guide you to do these kind of assessments. So if you have a fusion protein, for example, I think there is a risk if your protein undergoes proteolysis, for example, in circulation, that a part of the active site gets clipped, and therefore you lose efficacy. Therefore, it's very important to address this kind of early in research, because you want to bring a candidate to the clinic which actually is stable and doesn't show clipping in any part of the molecule. Similarly, I think amino acid modifications and other post-translational modifications can also have a severe impact on efficacy and immunogenicity, especially if they occur in the CDR region of your therapeutic protein, and therefore that also needs to be addressed properly before you bring a candidate in the clinic. Last but not least, for ADCs and other molecules where really very toxic molecules are connected to an antibody via covalent linkage to a linker, I think there are also questions on the metabolism, because the premature release of these moieties in circulation could also bring some safety concerns, and also question regarding efficacy. So all of these reactions we just briefly described show an impact on efficacy, safety, and immunogenicity. At the right end, we also have the pegylated proteins. I think for these we discuss at the end. I think they're a special case, but I think we also need to discuss the fate of the PEG in light of safety findings you obtain for pegylated proteins. So let's talk about one of the most common reactions, the proteolysis, which is the common reactions actually for fusion proteins, for example, in circulation. The in vivo proteolysis is species and disease state dependent. They can affect both exposure and the potency of your drug. We know that about that there are about 500 proteases expressed in human, I think even a little bit more, and there are species differences. So there's a clear need to understand the proteolytic stability risk of your drug before you move it forward. And we need this information to guide structure-activity relationships, to understand potential safety risk, but more importantly, if you have really a liability in your molecule that parts get cleaved, you need to try to engineer this out for the next, to improve the next generation molecules. So you really need this information to guide the protein engineering efforts. So what are the triggers to investigate proteolysis? One common one is, for example, mismatches between biological assay. And this is kind of elucidated on the left side here. So this is just a profile our colleagues from Janssen have published a few years back on dulaglutid. And you see here, if you just have, for example, monitor, or you have a biological assay just monitoring the stability at the stable part of the molecule, you will see always a pronounced exposure because it's not able to discriminate between the intact parent and potential cleavage products. But if you have more specific assays also for the active side, you probably see that these curves will drift apart over time. And this is a clear indicator that something is occurring on the molecule, and this is a trigger to start these investigations. Other triggers are unusual rapid clearance or a mismatch between the PK/PD relationship, where the expected pharmacological effect does not correlate well with the circulating concentrations. But we will revisit this in a minute. So how do we actually analyze these proteolysis products? So I think a typical assay platform to assess proteolysis is shown here. So if we collect our samples from animal or human, really the first step we do is immunoprecipitation to really separate the analytes of interest from all the matrix interferences. And what you do then is you inject it on your LCMS system. And for the LCMS, there's kind of different strategies you can follow. So the first, I would like to focus on the intact mass analysis or top-down analysis as is shown here. So for this, you inject your samples in your LCMS system. You do in your full scan, you see these nice protein envelopes for your protein, but also for some biotransformation products. And once you do the deconvolution, you get information, structural information about your biotransformation product, and you can probably conclude where the transformation has occurred in your molecule. This approach is very information rich. So it contains a lot of structural information, but I think for very potent biologics which require very low LOQs, for example, in circulation, it's probably not the best because I think it's not that sensitive. If you really want to have a more sensitive approach, this is shown on the right here. This is kind of the bottom up approach. So for this, you digest your protein with an enzyme after immunoprecipitation. So they get digested into peptides. You do a peptide mapping, and based on the modification, for example, of your epitope peptides, you can also draw conclusions on what modification have occurred on certain parts of the molecule, for example, the active site. This approach is a lot more sensitive than the intact mass analysis, but it contains less structural information, we must say. And then I think there's a middle way where you kind of reduce the size of your molecule by either reduce it chemically by cleaving the SS bonds or by cleaving light chain and heavy chain with certain enzymes. So you break your molecule apart to generate smaller fragments. And with these smaller fragments, you have a good compromise between sensitivity and structural information. And you can also extract a lot of information on your biotransformation there. This slide just shows you how you can apply this. So on the left side, this is a imaginative therapeutic protein shown. You can probably imagine it as a fusion protein. So you have a stable part, you have an active region, and in the active region, you have a label part, which is modified. So you can see if you have now different biological assays targeting, for example, the stable region and the active site, you can derive these concentration time profiles. So if you only look at the profile from the stable region, you probably see a pronounced exposure over time, because with this assay alone, you will not be able to discriminate between your parent and potential cleavage products, which occurred on the other side of the molecule. Only if you have specific assays which also target the active region, either this bottom-up peptide mapping approach or the top-down intact mass assay, or you have a bioactivity assay where you can really test the activity of your molecule against the target, then you see that these curves drifting apart. And as mentioned before, this is really a clear indicator that proteolysis has occurred and a trigger to do these biotransformation investigations. Other important modifications are the deamidation and the oxidation. So the deamidation of asparagine and isomerization, as well as the oxidation of methionine. These modifications, together with other modifications, which I don't want to discuss here, like the disulfide scrambling, duplication, etc., they can occur during the biosynthesis of your protein. They can occur during storage of your protein, but also after in vivo injection, meaning they can occur in vivo. And I think they're very, they have a high impact, as mentioned before. If, for example, if they occur in the CDR region of your protein, because then it can clearly reduce, change the structure, for example, reduce the binding, potentially increase immunogenicity, and have a big impact on your efficacy and PK/PD. So one way to check for this is by the so-called stress tests. So these stress tests are usually done in buffer. So my colleagues from Genentech were so kind to share with me this slide. So what they have done is they compared the results from a deamidation stress test in buffer to the in vivo results, and also the stress test results of an oxidation against in vivo results. And you see that for the deamidation stress test, you see a very good correlation with in vivo results, while for the oxidation stress test, the correlation between the in vivo results is not that great. So that clearly means that you need to develop some research and resources to develop better translatable assays, for example, for the oxidation. Now I want to talk a little bit about the tethered biotherapeutics, and especially the ADCs and the pegylated proteins. Proteins have been pegylated for a long time to increase the half-life. So the first pegylated protein which was registered was Adagen. In the meantime, there have been about 30 pegylated proteins been registered. For the ADCs, it's slightly different. Although the concept is known for, or the principal concept known for 100 years, I think only at the beginning of 2000, the first ADC was registered. And with the registration of Kadzilla and etc., a whole wave of new molecules came recently into the clinic, which clearly shows that this therapeutic approach has some potential, especially in the oncology space. So the chemical modifications, of course, are of concern, because you need to determine what happens with this chemical matter, where does it go, how it's eliminated, especially for these very potent toxins you connect to the antibodies. So I think the matters of concern are the drug release of the ADC before it reaches the target, potentially impact off-target binding and on efficacy, as well as toxicological findings. But nevertheless, I also want to highlight that we need to have a look at the modifications on the antibody, like described before. If you have some modifications, they can also affect the efficacy, and therefore it's for the ADCs, it's very important to really select the right antibody which you conjugate to the linker payload. And as mentioned before, these released biotransformation products, they can be still very potent. For some ADCs, it's even more potent than the original payload which you add on. And for the pegylated proteins, as mentioned before, you also usually need to at least look at the fate of the PEG and discuss this in light of the safety findings you usually have. So this slide shows you the general disposition of these maleimide conjugated payloads, linkers, so which you have for a lot of ADCs. So once the compound binds binding to the target is internalized, it's usually degraded in the lysosomes, and you end up with the linker payload and the terminal amino acid. This can be further metabolized, and in certain cases, this can be more active than the parent, but a lot of times it's also less active. And nevertheless, there's always a reason to monitor this. These cysteine conjugated payloads, they can also undergo retro-Michael reaction, so meaning they get deconjugated, and then the released maleimide, they can react with the sulfur for any protein it sees, and a lot of times this reacts actually with albumin. So if you have, for example, a stability assay where you monitor the intact ADC and the released payload, and you see the discrepancy between these two values, one reason could be actually that you have this albumin binding of the released maleimide payload in case you have a cysteine conjugated one. The modification of the linker is also important because if these maleimides get hydrolyzed and they open up and these ones are more stable, and actually use this principle for the newer generation of ADCs to actually stabilize the linker, these linkers to prevent the release of the payload and to prevent this retro-micro reaction from occurring. Last but not least, you can also have metabolism on the payload while it's still bound to the antibody. This is also important to monitor because in certain cases, if you have a hydrolysis or acetylation occurring there, this can also have an impact on the efficacy of your drug. For the pegylated proteins, I just want to say some also undergo similar reactions like these maleimide conjugated ADCs. You have multiple sites of conjugation. They're very heterogeneous. So I think for 40K pack, you have about 200 species in circulation. So it's really difficult to track all of them and look at them separately. It's not possible. I think a lot of sophisticated methods have been applied to do this. But in general, the perception is that all of them release the PAC as a major metabolite. Smaller, shorter chains of PAC can be further metabolized by alcohol dehydrogenase, while larger PAC is just eliminated as is, intact, mainly renally and biliary. PAC itself is perceived to be non-toxic, but a lot of times you see this vacuole formation in the kidneys, which is probably a natural mechanism to eliminate the PAC. So for a lot of pegylated proteins, you need to discuss the formation at the PAC, at least in light with the observed tox findings. So we propose to you actually to just a general decision tree to guide you which biotransformation you need to conduct, and this decision tree is just based on very basic questions. Is your therapeutic modified to conjugation or fusion? If you don't have any conjugation or fusion, you just need to do more or less the basic profiling. So you check you have CDRs, do you have the amino acid modifications or other PTMs? Is there proteolysis on your molecule and other glycolizations which are relevant? If you have fusions in your molecule, you will probably follow the same thing. Do the basic assessments, but put the focus really on your proteolysis investigations I've highlighted before. And if you have chemical conjugations in addition, then you will of course do this part to characterize the antibody, but in addition you will also look at the linker and the payload stability as highlighted on the previous slide. So where do we see this field evolving? So we feel that probably the complexity of these molecules will further increase because scientists always try to find more creative ways to drug formerly undruggable targets. With this, we need to make sure that we have the right tools in place. So I've shown for example, for the amino acid oxidation that the translatability is questionable at the moment, so better assays need to be developed. I think this is also the case for the proteolysis. Proteolysis is not as straightforward as some liver metabolism, for example, as it can occur in any tissues and in circulation. And usually you need to have a good understanding where your target is, to which tissue your antibody goes to really find the suitable in vitro assay for your molecule. So I think there is also need to develop better in vitro assays. With the increasing complexity of the molecules, also the complexity of the analytics and data processing is increasing. So we really need to have good tools which guide us and help us to elucidate these biotransformation products. I just want to highlight that these tools and software which are currently available are not really designed to do biotransformation data analysis on proteins. So a lot of companies try now to build their own in-house solution to address this need. We feel that this kind of fit-for-purpose approach, which we described here, will also remain there for the future. I think for some modalities like ADCs, the regulators try to put out some guidances right now. So there's some draft guidance available for ADCs. But I think for other modalities, we will probably not expect it in the future, because I think also the variety of biologics which are developed and are entering the clinics are quite diverse. So you cannot simply cover them with all kinds of guidances. We acknowledge that the proposed decision tree might appear a little bit simplified to some people, but of course we acknowledge that we emphasized a lot when we developed this strategy. We targeted biotransformation products which are potentially identified in circulation. So if you, for example, use novel delivery mechanisms, for example, conjugate antisense oligonucleotides to antibodies to have proper tissue targeting, you need probably to adapt this approach further in the future and adapt it, and probably refine this characterization strategy. So with this, I'm done, and I will hand over to Catherine.
Thank you, Marcus. So the focus of this sub-team was on the PK and biodistribution of therapeutic proteins, and not just for monoclonal antibodies, as you've seen, but for the increasing range of biological drug modalities under development. So during our discussions, it became clear to us that innovations in molecular design have given rise to more complex PK/PD relationships, which then require us to change the ways in which we characterize them. So now this slide shows a bit of a simplified generalization, but in years gone by, a lot of focus was on developing biologics that were generally targeting a single antigen, most often with an inhibitory, antagonistic mechanism of action, which meant that we wanted to achieve high receptor occupancy with the doses that we gave. But more and more over recent years, the industry's been developing biologics with more of a range of molecular formats and mechanisms of action. And so for some of these, such as immune agonists or peptide or antibody drug conjugates, which deliver high potency payloads, the receptor occupancy that we're needing is now much lower. And furthermore, we actually risk severe toxicity if we exceed even sometimes fairly low receptor occupancies. And then on the other end of the scale, for some drugs such as bispecifics, if we deliver doses that are too high relative to one or both targets, you can end up with a detrimental impact on efficacy. And you can see a sort of bell-shaped dose-response relationship. And when the target's located in tissues, systemic exposure may no longer be the appropriate input for these more complex PK/PD relationships. So that further adds impetus to think some more about biodistribution. So what we wanted to do as a group was to capture how we, as pharma companies, were rising to the challenges being presented by these new paradigms by looking at experimental and in silico tools used across the industry, and in particular focusing on what's important for key decision making during the preclinical phase of development. So an obvious first point of discussion was the in vivo biodistribution study, and there were differing views within the group on the utility of this type of study for biologics. But we could all agree that the insights gained from it rely on what we measure and how we measure it. So by that I mean which biological tissues and fluids we're interested in, and also whether we take samples or use less invasive techniques, and which analytical methods are used along with their spatial resolution, because all of these affect how we interpret the data and also whether we need to make any corrections to the measured values. For example, to account for the presence of drug within residual tissue blood, and also what to do if the labeling affects, for example, the PK properties of the native molecule. And those issues are sometimes similar to what we have for small molecules, but for therapeutic proteins, because of the different mechanisms affecting biodistribution, we also agreed that whatever the utility, whatever the views on the utility of the in vivo biodistribution study, it was definitely not something to be looked at in isolation, and we really needed to nail down what were the key processes that make up the net biodistribution that we observe. So for therapeutic proteins, PK and biodistribution can be influenced by any or all of the processes shown in this diagram. And these processes are governed by the biophysical properties of the molecule, as well as the binding kinetics and biology relating to either intended or unintended receptors and targets, as well as other components of the blood and tissue. And we demonstrate in the plots on the right how the observed PK profiles can totally change as a function of the interplay between these different processes. So here we can see two scenarios. If we look at the top graph for a biologic with low non-specific clearance, as the abundance of its specific target increases, the drug is more rapidly eliminated via target-mediated clearance, and we see increasingly steep PK profiles with shorter half-lives. But on the other hand, at the bottom, if the non-specific clearance of the free drug is rapid, then the binding of the drug to its target may actually result in a greater retention of the drug in the form of the drug-target complex, as long as the clearance of the complex is slower than the clearance of the free drug. So then the half-life of the total drug appears to get longer. And that's why for therapeutic proteins, it's important to think carefully about whether you need to measure free drug or total drug, and then what this means for your PK/PD interpretation. And what's also important is to be able to characterize, well, to quantify the contribution of the components making up the overall biodistribution and PK profiles. So what we then wanted to look at was how we can measure and look at these individual processes before piecing them back together again. And here we're showing a high-level overview of the range of approaches you might use to do that. But obviously a key question is, which data do we need to have, or which data can we consider interesting or useful to have at a given stage of drug development? So after discussion within the group, we of course decided that the simple answer is there's no simple answer. We couldn't really come up with a decision tree that covers all cases and modalities. But as a general rule, we certainly wouldn't advocate carrying out all of the in vivo experiments shown here on the right as routine during screening and discovery, for example. So what do? So as a first step, we want to think about what we know about the design of the molecule. And you can do this possibly using knowledge from existing molecules of the same modality or mechanism of action. And importantly, first we want to anticipate where our expected therapeutic dose range is likely to be within the receptor occupancy versus exposure curve. Or perhaps instead of receptor occupancy, another more appropriate marker of pharmacological activity, depending on the mechanism of action. And secondly, to truly understand how our particular molecule will behave in the body, we then need to ask ourselves the following key questions. What will be vital to our understanding of the drug response across dose levels and among different patients? i.e., what may cause inter-individual variability, and what may cause nonlinearity in the dose-exposure-response relationship? So thinking back to the simple case of the monoclonal antibody with the inhibitory mechanism of action, here on the left you can see the relationship between dose, exposure and response may look something like this. With the systemic PK of the total drug, you can work out at which dose range you saturate your receptor occupancy, and thus when your response plateaus out. But if your exposure at the target site is very different from that in the bloodstream, or if you have a number of biological processes contributing to actually get to your active drug component, as shown in this example on the right for a bispecific, then you may need some additional data and modeling to fully understand your dose-exposure response relationship. And there are some good examples in the literature of how these more complex cases can be modeled, and importantly, what sort of data is needed to do that. And we've highlighted some of these in our white paper. So some of the key learnings are that the more complex the model is, and the more parameters and assumptions you introduce, then you're more likely to need to actually combine data from multiple experiments, where the biological conditions in the assay vary. And actually, sometimes the PD response across experiments can't even be explained by the drug levels, and instead you need to measure or estimate the exposure of the drug target complex, or even use downstream biomarkers as the link across assays. And by doing all this, you can get a more robust characterization of your preclinical exposure-response relationship. And you can also investigate which are the key influential parameters using model sensitivity analysis. And the benefit of all this is that you're better placed when it comes to interspecies translation, because, of course, the main goal of all this is to make predictions in human. So ultimately, within the industry, we still have to be pragmatic. And this means that sometimes fit-for-purpose approaches can be appropriate for interspecies translation, even if more complex models are technically feasible. So we think there's a sort of sliding scale of mechanisticness which can be applied to translational modeling approaches. For example, in some cases, it may still make sense to use more empirical approaches. And that's, for example, when linear, non-specific processes govern the PK, where the time course of the drug in tissue matches the time course in the bloodstream, and where the extent of tissue partitioning is likely to be similar across species. So in those cases, systemic PK can be scaled using allometry. And there are lots of published examples of allometry which has been successfully applied for large molecules, often using non-human primate species, for example, from toxicology studies, but also even earlier with transgenic FcRn mice, which, as you saw in the survey results Prakash showed, about 60% of the responding member companies are now using these mice at early stages of development. Now, where we're more likely to need more mechanistic models are cases such as at lower doses with respect to the target levels, cases where target-mediated binding and clearance can have an impact on the overall PK, and where receptor occupancy varies across the dose range and thus may give rise to a steep exposure-response relationship. And also in the cases where the drug or disease-related changes in PD can have a feedback effect on the exposure at the target site. So here, quantitative systems pharmacology models, which include the downstream PD, can help to capture the relevant processes. And again, we've cited some good examples in our white paper. And now QSP models can be built starting from systemic PK models, but sometimes we'll need to predict what the actual exposure is at the target site, or indeed at other tissues of interest if we're talking about off-target toxicity, for example, which may vary across species as a result of differences in physiology. And a model framework that can do this is shown on the next slide. So here, an example of a physiologically based model framework is shown here, and in some ways it's similar to what a PBPK model for small molecules would look like in terms of the blood flow and tissue compartments. But actually, a number of things have been done over the years to adapt PBPK models to therapeutic proteins. So the addition of the lymphatic system, as this can be a key distribution component for large molecules, the particular extravasation and cellular uptake processes that are characteristic of biologics can be included, as well as intracellular processes such as FcRn binding and recycling, and also lysosomal degradation as a clearance mechanism. So as well as enabling cross-species translation via physiological parameters, these models have enabled predictions not just in human, but in patients with different disease states. And they've also been linked to PD and QSP models as well. And so the great thing about this type of model structure is that for more complex molecular formats, we've also seen cases where it's been further customized and on a tissue-specific basis to account for multiple targets, avidity effects, antigen shedding, release of conjugated drugs, and so on. So in our paper, we've included some references for some nice examples where this has been done within the industry. And going forward, it's particularly interesting to see how PBPK models for therapeutic proteins are evolving towards using in vitro inputs. So not just for the FcRn and target binding components, but for other processes such as non-specific cellular uptake. And so this can enable earlier use of these models prior to in vivo studies. And it's well worth a look at the references that we cite in our paper to see how they did it, how well it performs. So finally, we wanted to lay down the fruits of our discussions in the paper in the form of this table. And I won't go through all of it in detail here, much of it's already been covered. But if I could pick out a couple of key elements, I'd say that it's increasingly difficult to find a single in vitro or in vivo system which is able to accurately reflect the complexity of how newer modality biologics interact with their environment. So because of this, it's then getting difficult to make large-scale correlations based on in vitro or structural properties. Although we think that AI could ultimately help to do that, we'll need much larger data sets and also careful consideration as to whether we can, mix modalities within these correlations. So to sum up, I think I'd say that more than ever, we're needing like holistic approaches to contextualize and link all of the data generated during preclinical development of therapeutic proteins. And by understanding and quantifying the biological mechanisms behind their PK and biodistribution, and using tools such as mechanistic models, we can build a more robust characterization of the exposure-response relationships for both efficacy and toxicity, and then therefore a better basis for scaling across species. And with that, I think I'd like to conclude this portion of the webinar. Thank our group members and hand back over to Hongbin if I can.
Thank you very much, Kath. I would just quickly summarize our presentation. So on a very high level, as I mentioned earlier, our team members shared cross-company experiences and perspective and engaged in many scientific and in-depth discussions. That's what I enjoyed very much most in the last 3+ years. And we also had a strong collaboration, and it's really true hallmark of the ADME of Therapeutic Protein Working Group. And what we have hope is that our work will stimulate further discussions on ADME of therapeutic proteins in the scientific community, and together we can move the science forward. So here slide shows all the members that in the 3+ years, we had 40 members at various time joining the group. Some of them stayed through the 3+ years. So Catherine, those colleagues highlighted in green, Catherine, Prakash, and Kevin, Marcus, they are sub-team leaders. Brooke is the co-chair, as well. I also is the co-chair. I would like to thank Anthony from the IQ-TLG group as the sponsor for our working group, as well as the whole IQ-TLG leadership team. And also special thanks to Lana and Mahalia for their support in the last 3+ years and really help us to get to our finish line. And finally, we really appreciate all the support from the IQ Consortium and really feel privileged to be part of this great group and come up with recommendations for the therapeutic ADME of therapeutic proteins together. And at this stage, I would like to hand over to Brooke to kick off the panel discussion. Brooke, can you hear me? Yeah.
I can hear you just fine. Thank you, Hongbin. Thank you to our 3 speakers. Those were great presentations. I'm going to just give a minute for the panelists to come on the screen, and then I'll kick it off with the first question. Great. So the first question I'm going to direct to Catherine, and feel free to pass it along to any other members on your team if you'd like. But the question is, how do you decide whether a mechanistic or empirical model is better, especially if you've tested both and you end up getting different predictions?
Yeah, well, the simple answer, I suppose, is you need to think about what the model's being used for, really. I mean, if it's like for simulating repeated doses, for example, within the same species from which it was built, you might be okay using an empirical model just to kind of, get an idea of what your concentrations might look like under different dose regimens, for example. But for interspecies translation, then, as I've said in my talk, mechanistic models will tend to be better, particularly the more complex the modality is. But if you're seeing perhaps differences between the empirical and the mechanistic model, it tends to be a sign, you know, that you're missing something on the mechanistic side. Either that, or obviously you don't have the necessary confidence, or you've still got uncertainty or discrepancies in your system, you know, biological in vitro systems or your physiological model parameters. So it's useful in a way that it highlights that you might need to do some more investigations, look at some more in vitro studies or even in vivo studies. And then sometimes there's just things you can't do much about within the experiment side of things. You know, for example, your biological assay conditions can't possibly replicate the human environment, and you've used scaling factors. Maybe you need to, you know, look more at the scaling factors that you've used and look at the sensitivity and try and narrow down which ones might be responsible for your discrepancy, and then look at what you can do there.
Okay, great. Thanks, Catherine. That was a very great answer. Maybe I'll pass it over to Marcus for the next question. Where do you see the future of ADME support specific to biologics? What do you think our strengths are and any identified gaps the group can afford?
Actually, I would like to pass it along to maybe Michael or Shauna to answer that, as they're also part of the panel.
I think one of the things that I would point out is something you spoke to, Marcus, in your future perspectives, that we've done a really good job developing, you know, new mass spec technology, especially some of the, you know, hybrid Orbitrap instrumentation. But I don't think we've kept pace with, you know, the need for bioinformatic tools, especially as some of these more complex molecules, they incorporate both small molecule and large molecule components, and we really don't have good, you know, informatic tools to be able to look at biotransformation, for example, in the context of both large and small molecule modalities together.
Shauna, anything to add?
Yeah, I think that's a great point, Michael. You know, and as like the technology, both like the physical space of like the sensitivity and scanning speed of the mass spectrometers, plus more of the bioinformatics tools advance, I think that'll help us build more platform-based approaches. So it's not going to be a one-off per biotransformation event. You can have early screening assays up and running that would help us really evaluate these early on.
Yes,
and then
I
maybe
would like to add
one other thing that I could see is, so, you know, as we're starting to get into, you know, the industry, siRNA conjugates, you know, gene therapy, you know, new technologies that would help us do a better job of kind of assessing the ADME PK of those types of modalities too.
Yeah, and I would also highlight there's probably a need for better translatable in vitro assays if you want to reduce the number of animal experiments. We have seen that for certain reactions, the translatability is not that great. So I think that's also a need where we probably need to have more research to have the right tools.
Thanks, Marcus. I agree with you. I'm just going to pause. Does anyone else on the panel have anything to add to that question? Okay, great. The next question, I think maybe Hongbin, if you don't mind addressing this, there's a question about immunogenicity. And if I remember correctly, I think maybe that was out of scope for this working group, but could you bring clarity to that to the people on the call, please?
Yeah, I think immunogenicity certainly is a very important and very diverse as well as complex topic related to therapeutic proteins. I think immunogenicity certainly can impact PK, PD, etc. But within our team, we did not go in depth on the immunogenicity topic because we had a lot of topics to tackle. But at the same time, we're also aware that there are multiple industry consortium groups who have been working on immunogenicity topic. But certainly, I think immunogenicity would have some impact on the ADME of therapeutic proteins for sure.
Mm-hmm. Absolutely.
Yeah, I agree. Thank you, Ben. There's a question. I think this is more of maybe a clarity question to Prakash. It states responses in your presentation showed low percentage of characterization for absorption, but the BLA slide showed all companies studied absorption. Could you please clarify the inconsistency?
Thank you, Brooke. I think that's a question like what we have done in the BLA application. The slide, what we have shown is that it's combined pharmacokinetics and absolute bioavailability together, actually. So that's why we see that more concurrent conducting absorption studies, but we combine that. Where in the survey, what we asked is really specifically asking you have done anything to understand the mechanism of absorption. If the drug is, like if the therapeutic protein is administered in non-intravenous route. So there is a difference slightly. There is not the same one, basically the presentation twice. So I would like my colleagues Kevin and Jennifer to add on if they have any additional thoughts.
I'll just add on. So in our table, we put the PK under absorption because that's kind of where the location is in the BLA. And so that's why we left it kind of in that column for the table. Because that's what the guidance stresses to for where to put the PK data.
And one of the things we discussed was actually the structure of the BLA. We also see this in other regulatory documents. May need refreshing when we think more about biologics compared to what they originally were put together for small molecules.
John, do you have additional thoughts overall in the manuscript obviously, or anything else? Looks like not. Just go ahead, Brooke.
Let me unmute. That's fine. No problem. We'll move on to the last question here. And this is a question for the group. Maybe this time we'll start off again with Catherine, and we can go around the circle just to close out the discussion. They're curious, do you have any key learnings from the manuscript that you did not share in the presentation that you would like to highlight to folks?
Did we keep any secrets back, you mean? No, I mean...
yeah, I don't know. Maybe they think that we have some supplemental data we're not sharing, but I think it's a good question. If maybe just some closing thoughts even would be great.
Yeah, I mean, we obviously, there's a couple of things. The manuscript itself, it's a mini review in the, by definition, the journal definition. So we were all limited by word count, which is always the case, of course, but there's obviously so many more things we could have included. So of course, I'd say we were limited in the references we could cite, and would have loved to have added some more case studies, but hopefully we've managed to cite, the key pieces. There's obviously a lot more reading everybody can do. Yeah, and then of course, the discussions that we had within the context of the working group, we of course couldn't, put them all into the paper, but within our group in particular, yeah, it was pretty enriching seeing what other companies did. There were a lot of similarities, some heated debates on the utility of the in vivo biodistribution study for biologics, which is obviously something I mentioned in my presentation. Some thought it was completely useless and all we should be looking at is, biology. Some people thought it was still useful, but in careful consideration. So yeah, I think there's more to be said on that probably. Gerard was a member of the group as well. Gerard, do you have anything else to add there?
Yes, thank you, Catherine. Well, you have to keep in mind that the discussions that we had in the preparation of this paper took place during the pandemic. So we had monthly sessions. And for me, the interesting thing was that we all had a very open mindset in our discussions. We shared so-called maybe secrets. We had very open discussion, especially on this topic of PK, of therapeutic proteins and biodistribution. You could write complete books on that. And we had a limit in word count, and we had to clearly define what we wanted to discuss in our paper. And that worked all quite well in those virtual meetings in which we didn't meet in person. So what I really liked was the open mindset, and that we really made progress in our thinking. I learned a lot in this kind of discussions.
Great, thank you, Gerald. We just have 1 minute left. If anyone else wants to add any closing thoughts, feel free.
Maybe I can just, okay, go ahead, Prakash.
So we try to summarize the 3 years of our work in 15 minutes. There is a lot of components are presented in the manuscript. Please read through the manuscript and yeah, reach out to us if you have any questions or comments.
Great, thank you, Prakash. And maybe I'll pass it back over to you to Hongben for any closing comments on the working group.
Yeah, absolutely. I mean, come back to the secrets. I don't think we hold any secrets in our paper or presentations, but I do hope that colleagues in the audience open up your secrets, because I think the ADME of Therapeutic Proteins is still emerging science. So the more you publish your case examples, the more it will enhance and enrich this part of the science, and so that we can move the science together. And finally, I would like to thank everyone, all the panelists, all the presenters, and all the 40 members who have been with us in this journey in the 3+ years. And it has been great fun, and we also achieved a lot. So a big thank you to every one of you. So great job.
Thank you, Hongben, for the great leadership.
Yeah, you're welcome. Thank you so much. You guys made this.
Okay, with that, I'll go ahead and end the webinar. Thank you all for joining us on this Friday afternoon or morning and afternoon. If you have any questions after the webinar, feel free to send them to the IQ Secretariat. I will be in touch with you with a copy of the slides and a link to the recording. And I hope that you all have a great weekend. Thanks to all of the panelists, speakers, and moderators for your hard work. And we look forward to any upcoming webinars on this emerging topic. Thanks very much. Have a great weekend.
Okay, thank you everyone. Bye-bye.
Thank you. Bye.
Okay, bye-bye.